Alrighty guys, I'm going to make a whole new video here because we had internet issues today which caused Zoom to kick off and not come back on, and also caused some projection issues where our large screen was concerned. So lesson 32, nucleic acids and energy in living systems. Alright, so what are nucleic acids? Nucleic acids are made of a phosphate sugar chain with nitrogenous bases connected to sugar units. So if you look over here, what you have is a phosphate sugar, and these are the nitrogenous bases, or what we're going to refer to as our nucleic acids. This right here is a model of deoxyribose. Now, deoxyribose is what we're dealing with right around in here, and deoxyribonucleic acid is where we get DNA. So when you combine this deoxyribose here with your nucleic acid, you get deoxyribonucleic acid, which is DNA. So DNA uses 4 bases and forms a double helix by specific AT and GC pairing. Replication can take place only when the helix splits apart. So if you look over here on the right, you have A and T, and you can see they're like puzzle pieces that fit together. And then you have G and C, and once again, they're like puzzle pieces that fit together. And we did go over this a little bit before lab when we did our DNA base pairing game, where you had to find the words to make the funky sentences. Over here are some models of our nucleic acids or our nitrogenous bases. So we have thymine, adenine, guanine, cytosine, and uracil. Uracil is only present in RNA, and in DNA, you have thymine where you would have uracil in RNA. So if you look here, you'll notice that uracil and thymine have a very similar structure, with the exception of thymine having this little extra guy over here. And the part of thymine and uracil that do the bonding isn't impacted by that one little spot. So when you're dealing with RNA, anywhere you would have thymine, meaning whatever's paired with adenine, becomes uracil in that case. So DNA base pairing, we have 2 types of base pairs. Our pyrimidines are our thymine and our cytosine, and our purines are our adenine and our guanine. And the base pairing bonds as shown here. Your O and your H and your N, bonding your thymine and your adenine. And in this case, your H and O, your N and H, and your O and H, bonding your cytosine and your guanine. So here's a graphic of how DNA replicates. So what happens is you have DNA that looks basically like a ladder, right? It's just like a ladder that's twisted. And then it gets unzipped, just like you would imagine a zipper unzipping. The exact same thing happens. You can see that happening right here. And then you have DNA polymerase that moves along the chain of this unzipped ladder. And as it does so, it places the appropriate nucleotide for a pair. It places that right there, and basically that's how the chain gets duplicated, kind of like twice at one time. And this says the arrangement of base triplets on DNA encodes genetic information by dictating the synthesis of proteins. All right, so if you remember from our DNA coding lab where we made words and then the words made sentences, you needed 3 base pairs to create a codon. A codon right here is going to code for a specific piece of information that will allow for the transcription with RNA, mRNA, and this is how we create proteins. So it's kind of like the words, and when the words go together, they make a sentence, and the sentence makes sense. And that's basically like making one protein. So then now you have a sentence that makes sense, but in order to put it in context, you need a paragraph. So that's how you get a gene. You get several of these sentences together and you make a gene. A gene is going to code for a specific protein and tell the body basically what to do with it and where it goes. So this is a great graphic of how DNA transcribes to protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So gene technologies involve working with DNA and include DNA fingerprinting, cloning, and recombinant DNA. All right, so this over here on the left, this is DNA fingerprinting or DNA profiling. And this here is a graphic of what is typically done in a lab in a gel. It's like a little block of gel, and it's called DNA electrophoresis. And basically, what you do is you take a sample of something known. In this case, the victim's DNA is known. So you have a victim at a crime scene and you're able to collect his or her DNA. So that's your known. And then you have your unknowns. And in this case, we have DNA that was collected from the crime scene, 2 suspects, and a victim. When you run the DNA through the electrophoresis process, it produces a series of bands, and the bands produce a unique pattern. So this shows the DNA for the victim. And if we look over here at the crime scene, there are 2 samples from the crime scene, and we don't know who they belong to. But the second sample that says crime scene 2 is completely identical to the victim. So we know this is his or her sample at the crime scene. We also have another sample from the crime scene, though, that does not match the victim. What it does match, however, is suspect 2. And this is basically how police and scientists solve crimes using DNA profiling. Now, cloning was a big thing right here in 1996 because Dolly here was the first mammal cloned. And this happened right when I was in college, right when I was studying this exact process. So it was a really hot topic for discussion, as you can imagine. So basically what happened here is in the UK, they took this Scottish blackface sheep and they got an egg from her. She was an egg donor. Once they had the egg outside of her body and in vitro in a solution in a lab, they went through a process called enucleation, where they removed the nucleus from the egg. Now, meanwhile, they had a different species of sheep called a Finn Dorset. And as you can tell, they don't look the same. So they removed mammary cells from this sheep. So these are not reproductive cells, these are not eggs. They removed cells from her udders. And they took a cell from her udder that was a good looking, healthy cell, and they combined this cell from her udder with the egg from the black-faced sheep. So essentially what happened is the genetic information located in the nucleus of the mammary cell entered this so-called empty egg and created a new nucleus. So that egg then continued to develop through its embryological processes, getting to the first major embryological milestone, which is a blastocyst, which is a point when you've got basically it's like a bubble. This is a cross section, but it's like a bubble of cells with a little clump on one side. So they took this blastocyst and implanted it into a completely different sheep who was a surrogate ewe. A ewe is a female sheep. Put it right into her uterus, and then 5 months later, she delivers a lamb that they named Dolly. Dolly is genetically identical to this Finn Dorset. So this sheep donated the egg, this sheep gestated the offspring, and this sheep is identical to this sheep. It is more like a twin than an offspring. So it was a big deal when the news about Dolly came out, because it brings up lots of ethical questions, like, what else do we clone? Do we clone people? Do we, is that a bad idea? Do we clone our pets if we really loved a dog? And do we clone that dog later so we can have it again after it's passed away? All kinds of weird ethical questions come up. It's a fascinating topic. Now, recombinant DNA. Oh, and just so you know, cloning mostly since then has been used to create tissues that can be used to replace unhealthy tissues or missing tissues in patients that say have a major burn or injury and they've lost skin, or they need other types of cells in their body that can't regenerate. They can use cloning for that. So recombinant DNA has applications including making plants more pest resistant, developing vaccines, creating insulin and growth hormones. And this is just something I want you to know exists. So this is a graphic that shows you a process where you might use recombinant DNA. So this right here is a mitochondria, and mitochondria are the powerhouse of the cell. So we have two main types of energy production / storage that are complete opposites of one another. Photosynthesis and cellular respiration. And we've talked about this before. But in green plants, they use solar energy, carbon dioxide, and water to synthesize glucose in photosynthesis. That's what happens in photosynthesis in your green plants. And then the reverse is respiration, which is when you break down the glucose into carbon dioxide and water and you get energy from the process. And for the sake of understanding how cellular energy is produced, you get about 38 ATP ions per glucose molecule. That doesn't matter for you to know right now, but ATP is a process of cellular energy that we'll talk about more in biology. So in this case, with your plant, you take in carbon dioxide and water and energy from the sun, and you produce sugar and oxygen. The oxygen is released as waste into the atmosphere, and the sugar is stored in the plant. Okay, now, just so you know, the trees on our planet obviously do make oxygen for us, but they are not the primary source. The primary source of oxygen on planet Earth is from phytoplankton, cyanobacteria that live in the oceans. So last year, I think it was, when we had fires in the rainforest, and people were panicking, and they should have been, because it was a pretty terrible thing to happen. But a lot of people were concerned that we were burning the, air quotes here, lungs of the planet, and that we would suffer greatly for it in terms of oxygen availability. But truly, the rainforest produces a lot of oxygen, but it nowhere near as much as the oceans, the organisms in the oceans do. So then we also have the opposite process. So here we have sugar stored and oxygen released as waste. So in this case, we're going to take sugar and oxygen. So this is you eating. And if you're eating not something that's a green plant, maybe you're eating meat, I bet you the meat somewhere along the line ate meat that ate a green plant, or it ate the green plant itself. And then oxygen, which is what you breathe in. And then that process is going to produce carbon dioxide, which you breathe out, and water and energy. And this is how it looks if you compare it at a cellular level The chloroplasts are where photosynthesis takes place, and these are in your green plants. And the mitochondria is where cellular respiration takes place in animal cells. So this is just a graphic I want you to see, but I don't want you to worry about it yet. This is for next year mainly. So through the release of energy during the breaking of this third phosphate bond involved in here, ATP fuels life's processes, motion, synthesis, and transport. So we have this is a cell, and inside the cell, it shows you this big mitochondria. All this is like really not proportional at all. But you have a Krebs cycle, and then you have glycolysis, and then you have electron transport. So basically, you're going to take in your glucose, it's going to change to pyruvic acid. That's going to come down here. You're going to have a Krebs cycle going on, which we'll learn about next year. And then you get electron transport, carbon dioxide going out of the cell, water going out of the cell, and these little guys, ATP, being produced. And those are your little sources of energy. So DNA phenotyping. We talked about this today, and for those of you who weren't here, the actual conversation was missed. So I want to be sure I go over it with you, and this was your homework. So Parabon Nanolabs is the company that is doing most of the research where DNA phenotyping is concerned. So it says that since 2018, it's solved more than 120 cases with the help of their genetic genealogy and phenotyping methods. So here's how genetic phenotyping works. You get a sample from a crime scene, like we showed in the previous graphic, or maybe you get a sample from a body that is unidentified. And maybe that body is decomposed to the point where you can no longer see a face for identification purposes, or you can't see a skull, or maybe it's just a skull or skeleton, or maybe it's only a part of a body. Maybe you only find an arm bone and you're wondering who it belongs to, right? So what they do is they can take DNA either from a crime scene left behind by a perpetrator or from an unidentified human remains. And in this case, in the middle here, what they did is they had some unidentified remains, and they were able to take DNA and put it into this database where they have all kinds of information about what genes produce what proteins that present in what way when you look at someone's physical appearance. So they're able to do it by percentage. So in this case, they'll say like hair color, not red, blonde, or brown. And there is a 100% chance here, and it's 89.3, right around here. This is where they're saying you're likely going to find the hair color. And they're saying there's an 87.4% chance that you're likely going to find the eye color around here and the skin color around here. And then freckles. There is, it says right here, 22.20, 90.6% confidence this person has no freckles. So they put all this information into a computer, which creates a composite sketch, but it's a digital sketch. So it's kind of like when you've seen crime scene sketches before, and they put up an image on TV and they say, have you seen this man, or have you seen this person? And it's drawn from witness description. This takes all of the subjectivity out of it, and it makes it very data-based. So this is the image they came up with based on the remains they found. And this is the actual victim. So as you can see, that's pretty close. They did really well in this case. And once they were able to identify who the remains belonged to, they put a face to it, then they could put a name to it, and they could catch this man who it says accused of killing her and dumping her in the woods. Now this one is interesting too. In this case, we had a victim and a perpetrator that was unknown but left DNA at the crime scene. So again, they go through this whole process and they completed this composite image. Well, what they can't tell necessarily is age. So they're looking for a guy that looks like this, but they're presenting him at about 25 years of age because that's their choice of, I'm not sure why they choose 25, but I guess it's harder to make them look older with accuracy or younger with accuracy, probably where their data points are located. This is the actual perpetrator. And you can tell, I mean, even though this man is much older than his composite, that looks a lot like him. Also, if you happen to know this man when he was 25, you'd go, oh, look, there's old Bob again. And yeah, we thought he was up to something shady, and he definitely was. And as you can tell, He was also arrested in the murder of that young lady right there. So this is a brand new and evolving technology. I expect we're going to see a lot more of it in the future. It's absolutely fascinating. One other thing they've been doing lately that doesn't directly involve DNA, but is involved with chemistry, is they are able to take bones and most specifically teeth of unidentified victims, and they can narrow down the region of the world in which they grew up or where they lived most recently based on mineral deposits in their teeth. It's pretty crazy. So here is a little bit more about DNA phenotyping.
I'm Ellen McRae Graytack. I'm the Director of Bioinformatics at Parabon Nanolabs. And we do DNA phenotyping, so predicting a person's appearance, ancestry, all that stuff just from a DNA sample. We start from what we call genotype to phenotype data. So that just means a big database where on thousands of samples, we have both information about their phenotypes, like say eye color. We have, are their eyes blue, green, brown, and we also have genotyped SNP data. So we have got about 1 million SNPs on each person and their genotype, so that's their AA or are they GG at each site. And what we want to know is which of those SNPs is significant for eye color. So we basically look through at each individual SNP and score how well does this data associate with the phenotype data. If people who have an AA at this SNP always have blue eyes, well, that would be a really strong association. It's never that simple, but that's what we're looking for. But the thing is, it actually gets much more complicated than that. If you look through the literature in genomics, you find what they call missing heritability, which means there are these traits that we know are genetic. They pass through families. They're very clearly heritable. But when we look across the genome, we don't actually find all the information to explain all the variation in that phenotype. So that's missing heritability. We're only explaining some of the heritability. And one idea for what might be missing is actually interactions among SNPs. So you might have a set of 5 SNPs that each of them individually has no effect on phenotype. But if you look at all 5 of them together, there are certain combinations of genotypes that have a significant effect. But if you've got 1 million and you need to look at 5-way combinations, that's way more than you can ever test. I mean, you would be doing calculations till the end of the universe. We don't have time for that. So what we've done is we've developed software that's called an evolutionary algorithm or a genetic algorithm. And so what it does is it starts from a random population, and we're still doing scoring. We're looking at this combination of SNPs and scoring how well does this predict that phenotype. If it scores well, it gets to survive and breed and mutate, and we get to look all around it at other possible combinations of SNPs. If it scores poorly, then it dies off. It doesn't get to get searched around. So in that way, we're trying to explore the important parts of the space while not wasting the rest of our lives looking at the entire space. So that's how we're trying to discover SNP associations that haven't been found before. So that's just the data mining. Then we build it into a predictive model. So we take those SNPs that we found and we say, okay, we know that AA genotype is highly associated with blue eyes. So in our predictive model, it would say, if I see AA, I'm going to give a higher probability to blue eyes. That's basically what the predictive modeling is. So for kinship, we also started from scratch. It's a similar approach where we also have a database, except now instead of eye color being our phenotype we want to predict, it's I have these 2 genomes and they're related at this distance. They're 3rd-degree relatives, for example. So we're taking the same approach, except instead of looking at each genotype, now you have 2 genomes you have to look at. And so what we're looking at is how similar are these genomes? And can we use that information to predict the relatedness? And so we've got a database that goes out to, there are actually 7th-degree relatives in there, as well as many unrelated pairs. And we're using this to predict 2 people's relatedness. And the value of that for forensics, it's twofold. It could be maybe the perpetrator of this crime is related to another perpetrator, a victim, a neighbor, you could test any pair of people that you suspect might be related. The other possible use is if you have remains and you don't have an immediate relative to test, you don't have a parent or a first sibling, but maybe you have a cousin and you suspect that it might be this person's relative. Well, now you can test their DNA and see, does it match the relationship of someone a third degree relative? Well, the challenge with faces is basically to describe a face in a way that can be predicted. So from our point of view, if you can measure something, you can predict it as long as there's some underlying genetics. So what we do is we take the face and turn it into just a combination of variables. How wide is the face? How long? How angular? How big is the nose? All of those can be turned into numbers that can then be predicted. So we're looking exactly in the same way that we did with eye color. We're looking for what are the genes that are involved in this? How can they be used to predict what this person's face like? This specific person whose face is somewhere along all of these variables, you know, maybe their face is wide and short or long and narrow, all of those things are somewhere in that face space, what we call it. We just need to be able to predict those variables. So when we use this for casework, what we try to do is emphasize both what we can predict and what we can exclude. So we don't just say this person has blue eyes, because a lot of the time, maybe this person has dark blue eyes. They're sort of on the line between blue and green, and it's very difficult to predict or even know, looking at them, are their eyes blue or green? And so that is going to hurt our confidence. So whenever we report a result, we say this person has blue eyes, and we have 75% confidence in that, for example, because maybe their eyes are green. However, maybe we can also say with 99% confidence, this person does not have hazel, brown, or black eyes. And that's what's really important for investigators. We can tell them, you don't need to look at the people in your suspect list who have dark eyes, because they really do not match this profile that we've come up with. And there's only maybe a 1% chance that this person has dark eyes versus a 99% chance that they have blue or green eyes. So that's what we try to focus on, is how can you use this information to narrow a list of possible matches. And so we've been using this in casework. We've done several dozen cases at this point where investigators come to us, say this, I've had this case hanging over me for years. I have no witnesses, and I just don't know who to look for. If they still have DNA from that case, we can do our genotyping assay, analyze that, and tell them, okay, you're looking for someone who matches this profile, and this vast majority of the people that you see walking down the street are not going to match that profile. So you need to focus in on this smaller subset of your possible matches. What we really want to see in the future, as this is getting out there, you know, like I say, now it's mostly that oldest, worst cold case. But you know, imagine now a crime occurs, and before you spend 5 years looking at every person around, if you narrowed it down to 5% of the population, suddenly, okay, I'm only looking for this blue-eyed person, and that's really going to change the efficiency of these investigations, and that's what we're looking for going forward.
So I have one more video to show you, and this is on virus replication.
Viruses. Viruses are infective agents too small to be seen with a microscope. They all have a similar structure, being surrounded by a protein shell called a capsid, which protects the viral genome. Inside this capsid is either viral DNA or viral RNA. Also present is the viral envelope, which is covered in attachment proteins. The viral envelope is made of lipoproteins. So how do viruses reproduce? Well, they don't quite lay eggs, that's for sure. Viruses don't have a way to reproduce alone. Instead, they need to hijack a host cell. To do this, they approach a cell, and that attachment protein we mentioned earlier interacts with specific receptors on the cell membrane. The protein and the receptor are complementary. The contents of the virus, such as viral RNA and the reverse transcriptase enzyme, are then injected into the cell by endocytosis. The reverse transcriptase enzyme copies the viral RNA, forming double-stranded DNA, which is then transported to the nucleus. The viral DNA is then integrated into the host DNA, and the new host cell assembles new viral DNA and the proteins necessary for the capsid and envelope. The new viral DNA and enzymes then leave the cell by exocytosis, constructing a viral envelope from the cell membrane of the host cell. Eventually, this will kill the cell, while the virus, well, they're free to infect other cells, and the whole cycle begins again.
Ah, there we go. So that's your lesson for today. And sorry we had internet problems. It was crazy, but hopefully that won't be an issue next week when we're doing review. So start your review, get it done. Don't forget your Unit 9 test, and I will see you next week.